The goal of this lecture series is to bring members of our outstanding faculty to different areas of the university so that we may all learn from their scholarship and their insights. Our university has campuses in Baltimore, Washington, D.C., Montgomery County, and the Applied Physics Lab in Laurel, Maryland. With 3,600 full-time faculty members, almost every discipline that one can imagine is represented across the 10 schools and divisions of our university. Even with this range of disciplines and the geographical distribution of our faculty and students, we see common themes around our great university. We have shared values of excellence, integrity, collegiality, collaboration. And we're driven by curiosity, a desire to advance knowledge, and a commitment to bring this knowledge to the benefit of the world. Our hope is that these lectures will provide opportunities to share the riches of our talent across the schools and divisions of the university, and in so doing, to kindle creative processes in all of us and encourage the development of more interdisciplinary programs and interactions. We're especially honored that Dr. Greider will give the inaugural lecture in this series. She will be introduced by Dr. David Zapula and by Dr. Joseph Gall. Dr. Zapula is an assistant professor in the Department of Biology in the Krieger School of Arts and Sciences, and Dr. Gall is a member of the Department of Embryology at the Carnegie Institution and an adjunct professor at Johns Hopkins University. Dr. Zapula.
Okay, well, thank you so much, Provost Minor. As a member of the Biology Department's Seminar Committee, it's certainly a pleasure to be here today to help kick off this new Provost Lecture Series and to have Johns Hopkins' own Carol Greider here to present some of the work that led to her Nobel Prize a few months ago. I'd like to thank Provost Minor for his leadership and for his and his office's help with organizing this event. Carol Greider discovered the enzyme telomerase on a particularly auspicious Christmas Day in 1984 in the Blackburn Laboratory, where she saw striking data from an experiment she had performed. It turns out that we all received a gift on that Christmas Day as a result of this finding. Carol has proceeded ever since discovering telomerase with a steady stream of major findings while determining functions of this ever-fascinating enzyme and its role in maintaining genome integrity. Indeed, it is Carol's research on telomerase and telomeres that has led to her receiving a Nobel Prize, along with Liz Blackburn and Jack Shostak, and we'd like to honor Carol at Homewood campus for this awesome achievement today. Carol's Nobel Prize is a testament to her genius and energetic and fearless approach to doing science. However, her discoveries and their recognition also lifts many other aspects of science worldwide. And I thought I would just highlight a few of these at this opportunity. As Carol herself has pointed out, the Nobel for telomeres and telomerase is further proof that of the power of creative, curiosity-driven basic science that leverages model organisms. Telomerase was identified biochemically using extracts of a pond organism called Tetrahymena. It has thousands of copies of gene-containing DNA fragments in its macronucleus, each of which contains telomeres, which are added to it. So the logic was that if telomerase existed, this organism would be one that would have tons of this enzyme. They were right, and their careful and creative approach really paid off. This Nobel Prize is also a great thing for women in science. There are 40 female Nobel laureates, 10 of whom are in physiology or medicine, and 2009 is the first time that there were 2 female Nobel laureates in this category. Lastly, the Nobel Prize for telomerase and telomeres recognizes this major step forward in our understanding of cancer, as well as other diseases, and I suspect Carol will go into that in some detail today. So before I introduce Joe Gall, who will introduce Carol, I just want to interject at this point that if people afterwards want to come up to these 2 microphones with questions for Carol Greider after her seminar, that's the idea, and that will allow us to have the audio transmitted to everyone in the room, as well as to the video podcast that's being made of this event. Okay, so I will next pass the podium over to Dr. Joe Gall, but I want to say a few words about him as well at this time. So Joe earned the prestigious Albert Lasker Award for Special Achievement in Medical Science in 2006, as well as the Louisa Gross Horwitz Prize, which he actually shared with Carol Greider and Liz Blackburn. As perhaps Joe will expand upon next, he was the postdoctoral advisor for Liz Blackburn, who was Carol's PhD advisor at the time they identified telomeres. Joe has been at the Carnegie Institute here at the Homewood campus since he moved from his faculty position at Yale in 1983. He's been working assiduously at the bench ever since. In keeping with the theme for the creative use of model organisms to study major questions in biology, Joe is truly a master of the ad hoc use of the right organism for the particular biological question being studied. He also invented in situ hybridization with Mary Lou Pardue. If you enter a string of these three words into PubMed, you'll get about 100,000 papers that's cited. So without further ado, here's Joe Gall to introduce Carol Greider.
So I promise you there aren't any more after me before you get to Carol. It certainly gives me great pleasure to introduce my scientific granddaughter, Carol Greider. But before turning over the podium to her, I wanted to just elaborate very briefly on some of the history of the telomere research that David alluded to. And I'm going to break with a tradition that I've followed in my entire life. I've never shown slides in an introduction before, but I'm going to show you a couple of slides. So I'm going to do it very briefly. So the telomere research really all started with this organism tetrahymena, and this is a diagram of tetrahymena. It has two nuclei, a macronucleus and a micronucleus. The really unusual thing about it is the macronucleus has fragmented chromosomes, and some of those fragments only contain a single gene. And is that me or somebody else? Among those fragmented genes, one of the ones that we had isolated in my laboratory was the gene coding for ribosomal RNA, which you can see here. These are all uniform length, and of course, each of these molecules, this is the DNA electron micrograph, has two ends. And when Elizabeth Blackburn came to my laboratory as a postdoctoral fellow, we decided that she should look at the ends of those molecules and see what was there. And what turned out was that there was a very simple sequence there consisting of 6 nucleotides repeated over and over again. Now, at that time, we did not call this a telomere sequence. And that was probably a tactical error, but nevertheless, we didn't know that it was universal, and we had no idea of how they were formed. Later, Elizabeth Blackburn and Jack Shostak showed that these ends could be transferred to the ends of yeast chromosomes and functioned as telomeres, and that was the proof that they had a more universal property. And then the same sequence was found in other organisms later on. The really big surprise, though, came with Carol's work, where she showed that these ends are not replicated in the usual way for DNA, but they're added by a special enzyme, telomerase. And telomerase turned out to be a very special enzyme, as I'm sure she will tell you, containing not only a protein, but an RNA component, the RNA component carrying the template for the ends. Okay, so that was my brief history. Now we come to last December, and I just want to show you one nice picture of Carol. I think it's probably the last time she was relaxed since the Nobel ceremony, because everybody keeps asking her to do everything under the sun. Okay, so that was the recent history, and now I want to do very quickly the really past history, which maybe even Carol doesn't know about. And this ties her work into an even larger framework, and ties it also to Hopkins in a way that she may not be familiar with. So here, we're going to do a lineage analysis, since I'm a geneticist, among other things. And we're going to start with Carol Greider down here in 2009. And the yellow refers to a Nobel Prize. Okay, so Carol was a student of Elizabeth Blackburn, who got the Nobel Prize at the same time. Elizabeth Blackburn was my student. I was a student of a man named Donald Polson, who was a Drosophila geneticist. Donald Folsom was a student of Alfred H. Sturtevant. Now we're back to where at least the geneticists know who we're talking about. Alfred Sturtevant was a student of T.H. Morgan, who got the Nobel Prize in 1933. Now, T.H. Morgan was a graduate student here at Johns Hopkins in the biology department, and he was a co-graduate student with two other very famous people, at least to the biologists, a man named E.B. Wilson. Well, let me just say, for those who may not know, T.H. Morgan founded genetics in the United States, and he got the Nobel Prize for his work on showing that genes were on chromosomes. E.B. Wilson founded cell biology in the United States, and he was, as I say, a co-graduate student of T.H. Morgan. The third graduate student in that triumvirate was Ross Harrison. And you notice he's in yellow, but the yellow is faint. The reason for that is, remarkably enough, he was actually chosen for the Nobel Prize in 1917, but it was never awarded because of the war. And that you can verify by going to the records of the Nobel Foundation. But those three students were all students of a man named William Keith Brooks. And William Keith Brooks was the founder of the biology department at Johns Hopkins, and he was professor from 1891 to 1908. So, Carol, you probably didn't realize that you can trace yourself right back to the beginning of the biology department, and it's most appropriate that the scientific great-granddaughter of the founder of the biology department should be the first lecturer in the Provost series. And she herself is Daniel Nathan's professor and director of molecular biology and genetics, and she's going to speak on telomeres and telomeres, understanding basic mechanisms and their consequences. Carol.
Well, it's really an honor to be here, and it's nice to come home. And so you all won't mind about my fashion accessories, I think. I've got two microphones on, and I really should stand now behind the podium so that I am more modest. Joe said that he had just a few slides, but I was really very honored by his introduction. And what I would like to do here today is to try and do three things. First, I want to just give some background about the history of telomeres and telomerase, and you've heard some of that, the background that led to the Nobel Prize for the general audience. Then I'd like to tell you a little bit about the kinds of experiments that we've been doing over the last 25 years since the initial discovery of telomerase. And then finally, for the aficionados in the audience, I'd like to tell you just a little bit of a news story of some unpublished work, just in a few slides, just to keep you interested. So as you've heard, we're very interested in telomeres and telomerase. Telomeres are the ends of chromosomes. Here is the nucleus in the cell, and the chromosome is here. The telomeres are these protective tips on the ends of chromosomes, and they have to provide two really essential functions. First of all, they have to protect the chromosome ends. They protect the ends from any kind of enzyme that might be chewing away. They protect the ends from telomere-telomere recombination, and they protect the ends from any kind of end joining. So there are very specialized structures here. The second function that telomeres have to perform is to maintain telomere length. And you heard a little bit from Joe, and I'll tell you a little bit more about the maintenance of telomere length. So as Joe was alluding, the history of telomeres really goes way back. H.J. Muller was working in Drosophila, And he first found that the ends of Drosophila chromosomes had very specific properties. And he, together with Barbara McClintock, around the same time, who was working in maize, was trying to understand these protective functions of chromosome ends. And they coined the term telomere from the Greek, telo being end and mere being part, the end part of the chromosome. And what Barbara McClintock had found is that this special structure, when she was able to generate artificially a chromosome that had 2 centromeres, and in cell division, if one centromere was pulled to one end and the other centromere was pulled to the other end, that would generate a broken chromosome end. And what she showed is that broken chromosome end was unstable. It wasn't like a natural telomere. And when you have 2 broken chromosome ends in the cell, they would fuse together to generate another dicentric, generating this breakage-fusion bridge cycle, which she studied for many years, that gave us some insight into the fact that telomeres are somehow special compared to other parts of DNA or broken ends. The next part of the study you heard from Joe comes to the molecular era to try and understand what telomeres are made of. So when Liz Blackburn was a postdoc with Joe Gall, they used this organism here, Tetrahymena thermophila, and this organism, a single cell, contains over 40,000 chromosomes. And so if you want to understand the molecular nature of a chromosome end, you go to the source. And that's what Joe is very good at, is choosing the organism to answer the question. And so what they did was to define the first evidence for the molecular characteristic of telomeres. And they showed that these sequences contain tandem repeats of the sequence T2G4 repeated over and over and over again on the chromosome ends. So it was known, if this is a chromosome here, and these are the T2G4 repeats, it was known that the mechanism by which chromosomes are normally duplicated, if you imagine that mechanism of replicating every time the cell divides, and you go to the very end of the chromosome, it was known that the DNA polymerase can't copy the very end of the chromosome. And so it was predicted that telomeres would shorten progressively every time a cell divides. So how could this be true? How could you have telomere shortening every time a cell divides and still have organisms walking around on the face of the Earth? So there must have been some sort of solution to this problem of maintenance of chromosome ends. And when Liz Blackburn and Jack Shostak got together at a Gordon conference, they decided to do a very bold experiment. And this is an experiment that was published, the original experiment was published a couple of years earlier in Cell, but this is the follow-up paper in Nature. And what they did was to take a yeast mini chromosome, so we have here in light blue yeast DNA with a particular marker that they could follow, and to ligate on these T2G4 pieces from Tetrahymena, and to ask the question whether or not these telomeres would actually serve as a telomere in yeast. Now, these 2 organisms are in 2 different kingdoms. So this was a very bold experiment to ask whether or not the telomeres from one kingdom would function in the other. And so what they did was to transform this linear piece of DNA into yeast, and they found that not only was this maintained, that is, nothing was chewing away at the end, but as that linear piece of DNA was dividing in the yeast cells, what they found is that there was a little bit of sequence which was added on to the very end, which they then showed was actually yeast telomeric DNA. So this was very interesting. How could you have telomere repeats that now are in another organism and you're adding specific telomere repeats? So the yeast sequence is a slightly different sequence. So when you clone this and sequence across here, you can distinguish the tetrahymena from the yeast sequences. And so what they proposed, which is a very bold proposal at the time, and this is taken from their Nature paper, we proposed that terminal transferase-like activities are responsible for extending the 3' end of the GT-rich strand in yeast. This was bold because at the time there were models that were out there that proposed how telomeres might be maintained that involved a DNA recombination kind of pathway. You can imagine if you have telomeres that are all the same sequence, that one strand could invade, a shorter telomere could invade a longer telomere, could be elongated by conventional DNA polymerase mechanisms, and then you could get net telomere elongation. So this was one of the models that had been proposed, and what Liz and Jack were proposing was something different, where you have the telomere and you have it basically extended de novo, and then you can fill in the other strand and get net telomere elongation. So when I came to Liz's lab, I set out to test whether this is true. That is, specifically, I wanted to look for an enzyme activity that had this property of extending the telomeric sequences. So to make a somewhat long story fairly short, what I ended up doing is developing an assay where we used single-stranded oligonucleotides. So this is just a 24 base long oligonucleotide primer, P32 labeled DGTP and unlabeled DTTP, and initially very crude extracts from tetrahymena to ask, is there any evidence for an enzyme activity which would elongate that single-stranded DNA? And what's shown here is the kind of result that I got after a number of years of months of tinkering with the assay. And this was a 6-base repeating pattern that extended up to the top of this gel. And what we were able to show is that that 6-base repeating pattern corresponds to the addition of this GGGGTT, GGGGTT. And these Gs are in bold because this is the pausing position that gives you this nice banding pattern extending up the top of the gel. So this was very exciting to find an enzyme activity that would elongate telomeres. But then, of course, that then opens up what the next question is. How can an enzyme actually just add this sequence? Where does the information actually come from? So the next step was to find out how can you have a DNA polymerase simply synthesizing a particular sequence. And what we decided to test is whether the enzyme may have an RNA component. And in fact, that's what we showed, is that this enzyme has both the protein component as well as an RNA component. So that's when I went into the cold room for about 2.5 or 3 years and developed a biochemical purification. So these are just column fractions off of a number of different biochemical purification steps where we're following the enzyme activity across many columns. And then evaluating what RNAs, so all of the RNAs are labeled over here, and it turned out that there was this little faint RNA here, which after another 2 years we were able to show is actually the RNA component of telomerase, simply by cutting this out of a gel and sequencing it directly. So what we found then was that the telomerase enzyme solves this problem of the shortening during replication. And this is a little cartoon of telomerase. There's an RNA component with its very nice folded structure. And within that RNA component, there's a small region which can serve as a template for the addition of those telomeric sequences, so that it can get on to the end of the chromosome here, and that what telomerase does is to elongate that chromosome end, so that telomeres aren't a unique size. Rather, they're in an equilibrium. There's a little bit of shortening and some lengthening, and shortening and lengthening. Now, I point out here the two essential components of telomerase, which we imaginatively call the telomerase RNA, or TR, and the protein component, which was identified a number of years later by Joachim Ligner in Tom Czech's lab. And this is called TERT. And this stands for telomerase, TE, reverse transcriptase. It turns out that this is a protein component which has homology to reverse transcriptase enzymes, so that it can use this RNA and synthesize DNA onto the ends of the chromosomes. So I'm just introducing these because I'll use this term TERT and telomerase RNA later in the talk. So this is basically what telomerase does, and we're still very fascinated by the biochemistry of this enzyme. How can you really have a polymerase that's copying part of itself reiteratively? And this is still really a fascinating question. But what I'd like to tell you a little bit more about is some of the other work that we and many others in the field have done in the intervening years, which is to answer the question, why does telomerase matter? And it turns out that what we've found over the years is that telomerase is required for all cells that have to divide many times. And I'll show you two specific examples of that. The first is in cancer. If you imagine that there's a particular tissue where one cell has undergone a genetic change such that it can then go on and continue to proliferate and become a cancer cell, that one cell is going to divide many, many more times compared to the surrounding tissue around it. And it turns out that this cancer cell needs to solve the telomere problem and to have telomerase to be able to carry out these many, many rounds of cell division. The other area has to do with tissue renewal, tissue-specific stem cells. These tissue-specific stem cells have to self-renew, but they also have to give rise to cells that are going to divide many, many times to carry out tissue renewal. And I'll tell you that telomerase is essential for this many cell divisions that the cells have to go through for tissue renewal. So when we first started asking the question about why does telomerase matter, I was collaborating with Cal Harley, who was interested in a phenomenon called cellular senescence, where you take primary human fibroblasts and put those in culture. And it turns out that those primary cells will only divide for a certain number of times in culture. And this is just taken from a southern blot looking at human telomeres. And you see this distribution of telomere lengths here. So this is total human DNA probed on a southern blot with the human telomeric sequence repeat. And you can see that as these cells are dividing for increasing numbers of generation, there is a decreasing size and a decreasing intensity of these telomere repeats. So that was very interesting to us, and it turns out that these cells either lack telomerase or have very, very low telomerase, and the telomeres are shortening progressively due to this incomplete replication problem. We then went on, again in collaboration with Cal Harley, is to look in normal human population as people age. And if you look at mean telomere length at total white blood cells versus age of the donors, it turns out that there's progressive telomere shortening in the white blood cells in these individuals. And we were very interested in this. And at the time, we thought that maybe these cells didn't have telomerase, and that's why you have this telomere shortening. But what I'll tell you now, which will be a theme throughout the rest of the talk, is that telomerase is limiting, that the cell only has a very limited amount of telomerase. And although telomerase is active in normal white blood cells, the number of cell divisions these cells have to go through outstrips the ability of telomerase to elongate it. So you get this progressive telomere shortening. So this will be a theme that telomerase is limiting in cells. And then more provocatively, in the last few years, a number of people have done epidemiological studies looking at stratifying people into long telomere and short telomere and looking at survival. So here's an example from a paper that Cawthon published a few years ago, looking at individuals 65 to 75 years, stratifying into those with long telomeres and those with short telomeres. And this is the proportion surviving. So they suggested that there's a survival difference depending on telomere length. So this is very provocative, but what we wanted to know was why? What is the mechanism? What would it be that would generate a differential survival of individuals depending on telomere length? And so we've been working over the last number of years to understand the mechanisms of what actually happens when you lose telomere function. So the approach that we wanted to take, being geneticists, were to do a genetic experiment and to use laboratory mice to try and understand what actually happens when you lose telomere function. And so what we set out to do was to generate a mouse that lacked telomerase. So when Maria Blaska was a postdoc in the lab, she set out to clone the essential RNA component. So the small m means mouse, so this is the mouse telomerase RNA. This is present in a single-copied gene in the mouse genome. It's also single copy in humans. And we generated a mouse in which one of the two alleles was deleted for telomerase. So when you cross two such mice together, these heterozygous mice, you get, of course, the wild type, you get the heterozygote, and then you get the completely null animal. Now, these null animals were perfectly fine. We could find nothing wrong with them. And we called these the G1, or the first generation in the absence of telomerase, and you'll see why. Since we didn't see any consequence here in this first generation, what we did was to take these G1 null animals and breed them to each other to generate the G2, and then the G3, cross the G3s to get G4s, et cetera, all the way up through the sixth generation in the absence of telomerase. And I will tell you in a few minutes that there are phenotypes in these later generation mice, but there are not phenotypes in the early generation mice. But the first thing I want to show you is that when you do this kind of experiment, we wanted to know what's actually happening to the telomeres. And so we used an assay that was developed by Peter Lansdorp at the Terry Fox lab. Since mouse chromosome end fragments tend to be very long, they're about 100 kilobases in length if you look at them on a southern blot. And so it's very difficult to see a small change in a very large restriction fragment. So in order to see whether there was changes occurring in telomere length with these mice, we use this assay, which is a fluorescence in situ hybridization assay, thanks to Joe Gall. So what you're looking at here is mouse chromosomes that are stained. The blue is the DAPI stain. And the telomeres are hybridized with a telomeric repeat. And this has just 3 copies of these telomeric repeat. So the number of probes that hybridize to the end of the chromosome then will determine how intense the signal intensity of hybridization is. So you can correlate the intensity of each one of these spots to the telomere length distribution. So when we do that, if you look in the parents, you get this very nice frequency distribution, with this being the signal intensity and the number of ends that have that intensity. And this looks very much like that heterogeneous smear that I showed you of the human telomere lengths. Because there's some shortening and some lengthening and shortening and lengthening, you have this frequency distribution. Now, when we look in the G2, the G4, and the G6, you can see that the telomeres have lost signal intensity. And in fact, there are some chromosome ends in the population where we don't detect any hybridization. That doesn't mean there are no repeats on these telomeres. It just means that they've fallen below the level of detectability of our assay. But they're the shortest telomeres in the population. The other really nice thing about this in situ hybridization assay is that you're actually looking at the chromosomes, and you can see what's happening. So this is the metaphase spread that I showed you earlier of mouse chromosomes. If you look in the wild type, the G2, the G4, and the G6, there's a couple of things that we noted. First of all, when you look at these mouse chromosomes, you'll notice that they all look like these little boomerangs. And that's because mouse chromosomes are all telocentric. The centromere for all of the chromosomes is located all the way along one chromosome arm. And so in each case, the centromere is down here. When we started to look at these later generation metaphases, what we saw are chromosomes that look like this. And what this is 2 mouse chromosomes that have fused end-to-end through their telomeres. And what I told you was that what the telomeres actually do is to protect chromosome ends. So this is evidence that now telomere function has been lost. Not only have we lost telomere sequence, but we've lost telomere function. So this was very satisfying to see. So I told you I would tell you a little bit about what actually happens when the telomeres get short. And what we and others have found is that when the telomeres get short, at the cellular level, you have either cell death, apoptosis, or cellular senescence. At the level of the organism, the first thing that we see is a decrease in fertility. And we showed that has to do with apoptosis of the germ cells. You can look in the seminiferous tubules and see that those cells are undergoing apoptosis. There's also apoptosis in the white blood cells and in the gastrointestinal tract, all tissues where you have a lot of cell division and cell turnover. However, we didn't see any of these phenotypes in the early generations. And what that tells us is that it's not the absence of telomerase that's causing the phenotype. It must be the short telomeres, because the only difference between this mouse genetically and this mouse is that these mice now have short telomeres. If it were the absence of telomerase per se, you would see that in this first generation. So that would be a second thing. Telomerase is limiting, and it's the short telomeres that are causing the phenotype. So we've been trying to investigate and understand how is it that short telomeres would lead to apoptosis. And we and a number of other people in the field have come up with a model, and that is that short telomeres induce a DNA damage response. Illustrated over here is what happens if you have a double-strand break in the cell. If you have DNA and something causes a double-strand break, that double-strand break is recognized by specific proteins, and it sets off a signal transduction mechanism, which works through the p53 protein, and then you can either get apoptosis or senescence depending on the cell type. So the telomeres are long and they're protecting the chromosome ends and making them not look like a double-strand break. When the telomere gets to be very short, now it doesn't look like a telomere anymore. And that sets off a signal transduction pathway, which we know goes through p53 and results in either apoptosis or senescence. So we're very interested, and a number of people are working in the field to try and understand what are these things that are recognizing telomeres as short telomeres, and what is this signal transduction pathway that takes you to p53. But this is the way we believe the mechanism that occurs whereby short telomeres lead to cell death or cellular senescence. So I'll tell you just one brief story about how this plays out in the situation having to do with cancer. Our lab, as well as Ron DePino's lab, with whom we collaborated to make the knockout mouse, took this telomerase knockout mouse and crossed it to a number of tumor-prone cancer models. And in most of those situations, what we found was that there was a decrease in tumor formation. We then went on to try and study this in a little bit more detail to look at some of the mechanisms. And I'll show you work from David Feldzer looking at a particular tumor type where we could show that the short telomeres dramatically limit the growth of these cancer cells. So David was very interested in a particular transgenic mouse, and this is the emu-MYC mouse. So this is a MYC oncogene that's driven by a specific promoter that's B-cell specific. So mice that are transgenic for this MYC oncogene get B-cell lymphoma with 100% penetrance, and they die of that B-cell lymphoma. And so what David wanted to know was what happens when you generate mice with short telomeres. So he took the MYC oncogene on the heterozygous MTR background and crossed that to the MTR heterozygous mice and generated G1s. And of course, after generating G1s, he had to make the telomere shorter, so he continued to select for the MYC oncogene all the time breeding with the telomerase knockout future generations to generate the transgenic mouse on this background. And what he saw was really quite striking, and that is that there was a dramatic increase in survival when the telomeres are short. So what's shown here is the percent of mice alive versus the number of days. And the emu-MYC mice on their own that are wild type for telomerase is in the black curve. So what you see, here we start off with 100% alive. After 100 days, half of the mice are no longer alive, and they've all died due to B-cell lymphoma. Now we take the case here where the emu-MYC mice are null for telomerase, but the first generation. So these have long telomeres. And what you find is that the survival curve is really essentially identical. That is, the absence of telomerase per se has no effect. These mice are all dying of B-cell lymphoma. However, when you look now at the emu-MYC mice that have short telomeres, what you find is that there's a dramatic increase in survival in these mice. And when David actually went in to look at these mice, what he found is that there were little microlymphomas in these mice. So the tumors had initiated and started to grow, but then they couldn't continue to grow due to the short telomeres. So this is another example that it's the short telomeres, not the loss of telomerase, that blocks tumor growth. So the telomerase is absent here, but when you have short telomeres, that's what really makes the difference. And then the last thing that I want to tell you about is some of the newer work that has really taken off, and that has to do with the role of telomerase in tissue renewal and in age-related disease. And this really started from a paper that was published several years ago now from the Dokel group in London. Now this was a group that was interested in a human genetic disease that's called dyskeratos congenita. And the paper is entitled, "The RNA component of telomerase is mutated in autosomal dominant dyskeratos congenita." So they were a genetics laboratory, and they had a number of families that were where this autosomal dominant disease was segregating in the families, and they did genome-wide mapping and linkage analysis, and what they found was it was the RNA component of telomerase which was linking to their disease. So we got very interested in the role of telomerase based on this initial paper. And what we realized was that we had in our hands a very nice tool to be able to dissect what's happening in this autosomal dominant disease. So let me tell you just briefly about dyskeratos congenita. It has a very long and complicated name, but basically these individuals that have this disease die of bone marrow failure. So some of the clinical features are that there are skin and nail problems, skin hyperpigmentation, rashes, abnormal nail growth. And that's what gave the disease its initial name when the people would come in with the clinic with this triad of phenotypes. However, the mortality is due primarily to bone marrow failure, although I'll tell you at the end, pulmonary fibrosis is also a major mortality factor in this disease. Now, the bone marrow failure is very interesting because this is a particular tissue of high turnover where the cells have to divide many times. And so we think that this is an example of the cells coming down and the telomeres getting short as the cells are trying to do tissue renewal. But I'll tell you a little bit more about that. So one of the nice things about being at Hopkins is having the interaction with clinical colleagues. And we were studying our telomerase knockout mouse, and we were associated then with some of the clinical connections. And Mary Armanios, who at the time was a clinical fellow and now is an assistant professor in the oncology department, was studying this particular family that she calls Hopkins family 1, where there was autosomal dominant inheritance of dyskeratosis congenita. And you can see the autosomal dominance. So here is a grandmother and grandfather, and then the parents of the proband that came into the Hopkins clinic. And you can see all of the individuals that are shown in black had mutations, Mary showed, not in the RNA component of telomerase, but in this case in the protein component of telomerase. So the protein component of telomerase is leading to this autosomal dominant inheritance of dyskeratosis congenita. And one of the things that we were very interested in that Mary showed in her paper is that there was genetic anticipation shown in these family pedigrees. Now, genetic anticipation means that there is an earlier onset and a worsening phenotype of the disease with later generations in the family. And typically you will hear about genetic anticipation with people that are discussing triplet repeat expansion diseases in human genetics, where this term was coined. But this should also remind you of our telomerase knockout mouse, where we have a worsening of phenotypes with later generations. So we were very interested in the fact that Mary showed this genetic anticipation. The other thing that really intrigued us is that the affected people are heterozygous for mutations. In this case, it's human telomerase RNA or human TERT. Why would it be that heterozygotes would have this disease? So if you have autosomal dominant inheritance of disease, there are really two models that people think about. One is one is a dominant mechanism, where if you have one wild type allele and one mutant allele, somehow that mutant allele goes and inactivates the wild type allele in a dominant fashion. The alternative is that if you have one wild type allele and one mutant allele, having half the level of the enzyme isn't sufficient, and that's called haploinsufficiency. And so what I'll show you is we were able to use genetics to show that in fact it's haploinsufficiency in this inherited human disease. So we wanted to test whether this dominant negative or haploinsufficiency. And so what we had been doing for a number of years is trying to develop another mouse model which may mimic more closely human telomere length compared to what we had been using previously. What's shown here is a southern blot of a number of different mouse strains. And the strain that I was showing you, this C57 black, is a standard laboratory strain where we had made the knockout. And you can see on the southern blot probed with the telomere probe that there is a range from 100 kb down to 19 kb, and this is a pulse field gel to resolve these large fragments. However, there were several other species of mice, Mike Heeman, a graduate student in the lab, found, where the telomere lengths were very similar to human telomere lengths. And so we had been crossing our knockout allele onto this particular background, shown here the cast EI, which has significantly shorter and much more homogeneous telomere length, thinking that we could see phenotypes more clearly in this genetic background. And we realized that we could answer the question of haploinsufficiency with this mouse that we had in hand. So we bred this 7 times for 7 generations onto this background to have a congenic strain. And what we could then do was a variation on what I've been showing you before. But in this case, we cross these heterozygous mice, and rather than looking at the null mice, we look at the heterozygotes that come out. So the mice at no time have lost telomerase. They just have half the level of telomerase. So we bred the heterozygous generation 1, heterozygous generation 2, heterozygous generation 3, et cetera, all the way down. And Lingyang Hao, the student that was doing these experiments, then was able to show at the level of telomere length that there's haploinsufficiency. So what's shown here now is our quantitative fluorescence in situ hybridization. In black is shown the wild type telomere length distribution. So this comes from these metaphase spreads where we're looking at the signal intensity. So this is the signal intensity, and these are the number of ends. So we have this nice distribution, and we're focusing on the lower end of the distribution. So that's why this bar is particularly high, because it's just the rest of this distribution. So here's the wild type in black, and the knockout is shown in red. Now we look at the heterozygote generation 1, shown here in gray, and it's shorter than wild type. But more importantly, the heterozygous generation 5 is significantly shorter than the heterozygous generation 1. So these mice are maintained always with half the level of telomerase, and we showed that they actually have half the level of telomerase. And the telomeres are shortening progressively. I've told you it's the short telomeres that cause the phenotypes, not the presence or absence of telomerase. So when we actually look in these mice, we see all of the phenotypes in the short telomere mice that are heterozygous. And this then recapitulates the human genetic disease of autosomal dominant inheritance. So again, the theme here is that telomerase is limiting in cells. If you have just half the level of the enzyme, telomere length can't be maintained. So we want to look at what the consequences of this are. And of course, when you're breeding heterozygotes, every time you breed the heterozygote, you also will get out a knockout. So if we look at the actual survival of these mice, again, this is a survival curve, the percentage of mice alive versus the number of days. Here's the wild type. Now we look at the knockouts that come from heterozygous generation 3 parents. They have a reduced survival. A knockout from heterozygous generation 6 parent, even greater reduced, and a knockout from heterozygous generation 8 parent have a dramatically reduced lifespan. And these mice all have problems in their GI tract, the turnover in the cells, a high turnover, and significant problems in the immune system and the hematopoietic system. And that was published recently, so I won't go through all of that data. But what this indicates is that the inheritance of short telomeres decreases the survival. And it resembles the genetic anticipation that's seen in dyskeratosis congenita. So we wanted to ask the question then, because I told you that both the RNA component and the protein component of telomerase are both implicated in this autosomal dominant inheritance. All of our model system that we had been using up to date was looking at the RNA component knockout, so deleting this component. We wanted to ask what would happen if we were to delete the protein component. And there are a number of families that have mutations in either one component or the other. So this has important clinical implications in terms of would you expect similar or different disease if you have mutations in one component or the other. And what I'm going to show you is that actually there's identical phenotypes whether you knock out either the protein component or the RNA component. In part, this was motivated because there are a number of papers that have been published in the last 3 or 4 years that have indicated that the protein component has a number of other functions besides its role in elongating telomeres. So we wanted to look very carefully because if it's true that there are a number of other phenotypes associated with mutations in the protein component, we need to know about that clinically. So there are a couple of papers that suggest that the TERT component plays a role in the DNA damage response. Papers that suggest that it's a transcription factor that is involved in Wnt-related transcription, and other evidence, both chromatin and Wnt. So we wanted to look at this very carefully. And so the first thing that we did was to look at our long telomere first-generation mice, where we see no phenotype at all, and ask, what happens if the protein component is missing versus what happens if the RNA component is missing, simply doing a microarray analysis. So this is looking at 3 different independent animals, and what's shown here is the change in gene expression. This here is the level of the change in the gene expression, so anything, and it's a log, so anything over 2-fold expression is on this side of these curves. This would be increased and decreased. And here's the p-value on the replicates. So the quadrants that we're interested in would be this quadrant here, which would have be a reduction in gene expression with a significant p-value, or this quadrant here, which would be an increase in gene expression with a significant p-value. And when we look at the protein component knockout versus wild type, we see no difference. And also when we look at the RNA component, we see no difference, and when we compare them to each other. So we couldn't find any difference in gene expression at any significant level when either of these components are knocked out. We went on then to look more specifically at the DNA damage response. And this is simply quantitating the response to DNA damage in either wild type or the RNA component knockout. And we're irradiating with either non-irradiating or irradiating with 5-gray and looking at the phosphorylation of the p53 response. And you can see here nicely on this Western, and it's quantitated here, that we don't see any difference significantly between the wild type and the RNA component knockout, and likewise between the wild type and the protein component knockout. So we didn't see anything different knocking out the RNA or the protein component. But of course, we're very interested in a clinical phenotype. And so what's really interesting to us is what would be the phenotype of the mouse. So we went ahead and took this mouse, and by the way, this mouse was generated by Lee Harrington, where she had knocked out 3 domains of the protein component of telomerase. We took this and crossed it again onto this castaneous background to be able to look at the phenotypes, to be able to compare side by side what are the phenotypes of the RNA component knockout versus the protein component knockout. And so we can look at the null animals, and we can also look at the different heterozygous generations. And so when we compare now the telomere shortening in wild type versus the mouse I was showing you earlier, the RNA component knockout, here is an example of wild type versus the RNA component knockout, and the age-matched mice, the wild type versus the protein component knockout, there was really a very identical degree of telomere shortening. We can show haploinsufficiency for the protein component. This is now, again, is the wild type and the TERT knockout, and then here are 2 different litter mates that are the protein component knockouts that are second-generation heterozygous mice. And you can see that this distribution falls between the control here, the wild type, and the TERT. So we have the haploinsufficiency that we saw with the RNA component knockout. And then finally, looking at the survival curve of these mice, we look at the percent of mice alive versus the number of days. And here's the wild type shown here in red. Knockouts from heterozygous generation 1 parents have a decreased survival. Knockouts from heterozygous generation 2 parents have a decreased survival. This is as far as we've gotten so far. We're still breeding these mice. But more importantly, we've looked at the pathology of these mice. And one of the questions is whether or not there may be developmental phenotypes because the Wnt pathway is involved in development. And there was some suggestion that the number of ribs may be a readout for this Wnt pathway developmental signaling. And when we look in either the C57 black mice or the castaneous mice, we find all of the mice have the normal 13 number of ribs. So we haven't found any evidence for any developmental phenotypes in these knockout mice. And then in a blind study with Bak Karim, who's in the pathology department at the medical school, we would give him our mice and ask him, what do you see in terms of phenotype? And he would see testes, empty tubules, as we saw in the RNA component, intestinal villus atrophy, bone marrow failure, and this decreased survival that I told you about. So what we find so far is that the phenotype of the RNA component and the protein component are very similar. And again, this theme that it's the short telomeres that cause the phenotype, not the absence of the enzyme per se. So just in closing, I just wanted to point out that many of the diseases that are seen in these families with insufficient telomerase share features of age-related disease. So we see bone marrow failure, immune senescence, chemotherapy intolerance, pulmonary fibrosis, and that's really Mary Armanios' story, very interesting one, liver disease, and increased cancer incidence. And so what we think is that a combination of the initial telomere length that's inherited combined with the cell turnover with age may predispose to certain of these age-related diseases. This is not all age-related diseases. These are age-related diseases that have to do with increased tissue turnover. And so I just want to point out this graph that was published a couple of years ago by the Valdez group. And this is very similar to a graph that I showed you earlier, looking in the human population at telomere length versus age. But there's just a lot more data here. And what you see is that there's progressive telomere shortening in total lymphocytes with age, but this really gives you a flavor of the distribution of the telomere lengths in the human population. It's very heterogeneous, and this heterogeneity will have to do with both inheritance as well as environmental exposure. But our data suggests that those that may be on the very low end may be more predisposed to these specific diseases of cell turnover, and this may be an answer to the question stratifying those with long telomeres and those with short telomeres, and maybe they're subject to certain ones of these age-related diseases. So just to summarize what I've told you today, I've told you that telomeres are required for chromosome end protection. Telomerase is essential for telomere maintenance. Telomere shortening leads to cell death or cell senescence after many cell divisions. Loss of either the RNA component or the protein component result in the same phenotype. I've shown you that short telomeres limit tissue renewal, and suggested that telomere length may predict the onset of certain age-related diseases. And I just want to make the point again that fundamental discoveries can lead to clinical implications. We started here from this unlikely ciliated protozoan tetrahymena, just from the curiosity of understanding how telomeres were maintained, and what we've found has implication in a number of different human diseases. So I'll just stop there and thank my collaborators. Margaret Strong has done all of the mouse work in the lab, and Sophia was looking at the TERT knockout mouse, and our collaborators, Bach Karam and David Huso in pathology, and Mary Armanios in her lab in oncology. Thanks. Happy to take any questions.
Very exciting talk. So I just have two curious questions. So first of all, what do you think is a mechanism make these telomeres to be limited? So are they made at one time of one stage of development, and throughout cell division they kind of get diluted out, or what is the other mechanism?
So the really interesting thing is that telomerase seems to be limiting, and both components of telomerase are limiting. It's very curious. In most biochemical reactions, you might have one rate-limiting step. But both the RNA component and the protein component can give you haploinsufficiency in disease. And that does seem to be at a cell-autonomous level that these phenotypes are carried out. And curiously, so one thing that people have suggested is perhaps the reason that telomerase is so tightly regulated is that if you can escape and have more telomerase, that's a predisposition to cancer. It doesn't cause cancer, but cancer cells need telomerase in order to develop. However, it even is true in yeast. So Tom Check's lab did basically the same experiment we did in the mouse, but did it in yeast. And even at the level of a single cell, having half the level of telomerase, you can't maintain telomere length. So I don't know why that is, but clearly the normal amounts that are present, the cell cares a lot to have only that amount of telomerase.
Yeah, another question is really just out of curiosity. I mean, within any organism, do you think there are cell type specificity of the telomere length? And across the animal kingdom, is there any correlation of telomere length with the longevity of the animals?
So there's no correlation across the animal kingdom with longevity. So we're talking about telomeres playing a role in certain age-related diseases, and that's not all of aging. If you compare these mice that we have, the black 6 mice, they have significantly longer telomeres than humans, or also some of the other mice, and they don't have any difference in lifespan. So I think that lifespan determination is going to involve a lot of different pathways. There isn't going to be one pathway that determines lifespan. However, the short telomeres may play a role in certain of these age-related diseases, which can be life-threatening. So they're going to be one component, but not all of the components. And then you said whether there's tissue specificity. So there is evidence of tissue specificity of telomere length, and whether that has to do with when you look at a mouse, for instance, that particular tissue, how many rounds of cell division since the zygote did that particular tissue go through? So that's one question. But the other question is, are there different rates of shortening in different tissues? And we don't know the answer to that yet. We would like to try and get at that using this fluorescence in situ hybridization assay. And then more importantly, when a cell is responding to a short telomere, are there tissue-specific mechanisms that tell you how short the telomere can be? It was quite striking to us that in the testes, that seemed to be, the fertility was the first thing that went in the mouse, and that perhaps the testes is more sensitive to a short telomere because you don't want to be passing on chromosome fusions. And we never saw inheritance of chromosome fusion, so that just never happens in the germline. And maybe the germline is more sensitive to the short telomere. So these are all hypotheses. We don't know the answer. Anytime we answer a question, we bring up many more questions than we've answered.
Thank you.
Mm-hmm.
Hi. You said that short telomeres in humans were associated with an increased cancer incidence, and that seemed to be the opposite of what you saw in the emu-mic mice, where short telomeres seemed very protective. Could
It's
you
paradoxical.
elaborate?
It's
Okay.
very paradoxical because it's known in the human diseases, both in the dyskeratosis congenita and pulmonary fibrosis, that there's an increased cancer incidence in those families. And the increased cancer incidence goes with the tissue. That is, those with a lung problem have an increased risk of lung cancer. Those with a bone marrow problem have an increased risk of leukemia. And so we see this a little bit in the knockout mouse. We can see in the intestine when we take sections where there's basically no villi because the telomeres are so short that stem cells can't renew and there are no villi. We see these little microadenomas and they don't progress. Presumably, if something went wrong, they might be able to progress. We don't know the reason for this. Of course, there's two categories of thought. One is perhaps 99.99% of the time, when the telomeres are short, the cells die. Some cells escape. Do the cells that escape then go on and have genetic instability that then leads to cancer? That's one thought. Another thought is the tissue reprogramming that's occurring. The tissue itself is trying to self-renew, and there's a lot of cytokines and local environmental changes that are occurring there at that site. And if something goes awry there, that could lead in a more global level, maybe not cell autonomous, but that could also initiate the tumorigenesis. So we're very interested in that question, and we don't know what it is.
Hi. It's been suggested that the reverse transcriptase portion of the telomerase is of antiviral or retroviral origin. So I was wondering what your thoughts were on that.
The
TERT component has homology to reverse transcriptases. And if you actually follow back the phylogeny, and Tom Check's lab did a number of these phylogenetic comparisons, it actually seems to be very ancestral. It looks like an early RNA-dependent RNA-dependent RNA polymerase. So actually the thought is that perhaps the viral RTs came from telomerase. Now, when you get back with those early DNA alignments, sometimes it's hard to know, which was actually there first, but it's very, very ancient. But it does seem to be linking to RNA-dependent RNA polymerases.
So I have a question about the brain. Since there are some cells in the hippocampus that renew themselves, did you find any learning-dependent deficits in your mice?
We haven't looked for learning deficits. There is some evidence from Maria Blasco's lab that there is some telomere shortening. If you look in the SVZ, we actually started to do some of these experiments with Randy Reed, but then it was published by another group. And we didn't get to the level of phenotype. His argument was that you'd have to lose so much before you saw a phenotype in that particular tissue. But there is some indication that the turnover in the subventricular zone does lead to telomere shortening. Whether or not there's going to be any consequence of that, we don't know. And maybe it would be worth pursuing more, but I haven't continued that.
So another general question. I read in the New York Times that exercise can actually decrease aging by maintaining the telomere length, and I would love to hear what you think about that.
I think exercise is a great thing to do. We should all exercise.
Carol, might we turn the explanations around in relation to Zin's question and to Karen's question. Is it possible that the telomerase is relatively non-communicating with the rate of cell replication? That, in other words, cell replication is much more sensitive to the environmental parameters. So there is a constitutive level of telomerase, more or less, and cells that all of a sudden get a signal to divide faster lose the balance, and eventually the progenies then end up with less abundant telomerase and thus the telomere shortening.
Yeah, we don't look too carefully at the levels of telomerase. There are a number of papers out there which measure levels of telomerase in various tissues and in various cancers, but we really have focused on the telomere length.
Right.
So that certainly may be, but what the consequence is isn't going to have to do with how much telomerase is there.
understand.
So we haven't spent a lot of time measuring how much telomerase, but rather looking to see what is the shortest telomere and develop assays to
Right.
look at the critical thing that's causing the cell death.
But if one assumes that this idea might have some value, then one can take the experimental animal and slow down the replication rate of the cells or of the tissue and look at the effect of that, assuming that the telomerase level will remain constant. Nutritionally, for example, slow down the cell replication.
Yeah, well, that would be something one could look at.
It was very interesting. You said in one of your slides that the telomerase affects immune system, right?
Yes.
Could you be more specific?
So there's bone marrow failure, the hematopoietic system, but if you actually look quantitatively at B and T cells, there's a hallmark of immune senescence with the ratio of the B and T cells. In fact, the CD4 to CD8 ratio looks very much like that you see in the HIV and in immune senescence.
So
So
there
I
is
didn't
a-
go into a lot of that. Mary just published that, but we can talk more
And B
in
cells?
detail. Yes, also,
Less B cells?
there are fewer B cells. We didn't look at a lot of the different markers in terms of ratios. We only looked in the T cells at the T cell markers.
We can talk about it more.
Yeah, yeah.
Okay, well, so there should be refreshments outside, and we